Another technique that we can use, apart from kind of just looking at the amino acid sequence in the protein, is to use something called immunological techniques to compare the proteins and look whether proteins in different species look at how similar or how different they are. Now, in order to do these immunological techniques, you need to have a bit of a rethink back to 3.2, where we talked about the immune system and the immune response. And remember that fundamental idea that the fact that antibodies will respond to specific antigens on a protein of another species. Okay, so try and keep that in mind when we talk through this different method. So let's take the situation where we want to use immunological techniques to investigate which one of the following organisms, a chimpanzee, a baboon, a spider monkey or a dog, is more closely related to a human. Now we're going to do this by examining a protein found in the blood called albumin. Okay? Now what we have to keep in mind, as we've said before, is that if we look at albumin as a protein, we'd expect two organisms that were more closely related to one another to have a more similar albumin structure. Okay, whereas organisms that were more distantly related, their albumin structure would be more different, because remember their DNA-based sequence would be more different, so they're more likely to have put slightly different due to the change in the code, due to those mutations. Their DNA-based sequence would have put slightly different amino acids in the polypeptide chain, which would have formed that albumin structure. Now, when we have a look at this technique, you don't actually have to be able to recall the technique, but it's really good to understand what's going on, okay? So remember, what we're trying to do is compare albumin from us, albumin from humans, with albumin from chimps, with baboons, with albumin from spider monkeys and albumin from dogs, and see which we're most closely related to. Now, some of you will have already seen this little rabbit here and be thinking, well, why on earth do we need a rabbit in this process? Well, you'll see now. What happens is serum albumin is taken from the human. Now, serum is a bit like, well, it's sort of the same as blood plasma, apart from it's had the clotting proteins removed from it. But we can think of it as kind of a liquid part of the blood that doesn't have the red blood cells and white blood cells in it. So serum containing the protein albumin is taken from the human, and that albumin and within the serum is injected into this little rabbit here. Okay, now the rabbit sees that albumin, that human albumin, as non-self. Okay, and as a result of that, the rabbit produces antibodies that are specific to the collection of antigens that are on that albumin protein. Now we need to remember, albumin doesn't just have one antigen on its surface. Albumin as a protein will have lots of antigens on its surface. So the rabbit will produce a series of antibodies to all of those specific antigens that are present on the human albumin. Okay? What you then do is that you extract the serum. So you extract the serum from the rabbit. Now remember, that serum is going to be containing all the specific antibodies that were made to the human albumin. Now what we're going to do in a second is do something with those specific antibodies. Okay, so what you do is you then take the rabbit serum. Now remember that rabbit serum is containing those antibodies to the human albumin, and you mix that with the albumin from a dog. Now, so here's our dog here. We'd mix it, the serum from the rabbit that's containing the antibodies to the human albumin, and we're going to mix them with a sample of serum albumin from the dog. Okay? Now, any of those antibodies that were produced by the rabbit that correspond to antigens in the dog serum, i.e. antigens on the dog albumin, they will then join together and form what's called a precipitate. Okay? And what you would do is that, excuse me, you'd then repeat that process for the other organisms that you wanted to compare. So you'd take the rabbit serum containing the antibodies to the human albumin, and you'd add that to a sample of albumin from the spider monkey. Okay, then you'd do the same and you'd add it to a sample of albumin from the baboon. Then take it again, add the serum containing the antibodies to serum containing albumin from a chimp. And what you can see is that you get different levels of precipitate. This green liquid shown in here, okay, represents the precipitate. And you can see that you get different levels of it. Now, what we can then see is that the greater the number of similar antigens, okay, the more precipitate is formed. Okay, and so the more precipitates formed, because they had a greater number of similar antigens, that tells us that the species are more closely related because their protein structure must be more similar. If their protein structure is more similar, they're going to have similar shaped antigens, and hence they would form more precipitate with these antibodies that were formed to that antigen. And so in terms of, sort of questions that you would be asked, you would just be asked to sort of look at the amount of precipitate formed, or it might give you a value for the amount of precipitate formed. And you just need to keep in mind that the more precipitate that's formed, that tells you that there were a greater number of similar antigens on the albumin from the chimp and albumin from the baboon, and hence you get a greater amount of precipitate formed with those antibodies that were produced by the rabbit to the human albumin. And it's just another way of sort of saying, yep, these organisms are more closely related, these organisms are more distantly related. So this might be some data you might get in an exam question. And here we can see we've got serum from rabbit containing antibodies against human albumin was added to serum from the species below. So it would be added to serum albumin from the chimp, from the gorilla, from the orangutan, from the gibbon, and this time it was from a lemur. And the results show the relative precipitation level. Okay, so obviously we'd get 100% precipitation when we compared it to the human antibodies, to the human albumin, because obviously there would be, that's what they were originally raised for, and then you get a relative amount to that 100%. Okay, so take a minute, just pause the video and think about the two questions that follow. So, if we have a look at the answers then. So, which two primates does the immunological study suggest are the most closely related? Well, the most closely related are going to be these guys here. And we can tell because, relatively speaking, they get the same level of precipitation when they are combined with the antibodies to the human albumin, suggesting that their protein was most similar to each other, and hence, because their proteins are most similar, they are most closely related in evolutionary history. The primate that the study suggests is the nearest relative of the orangutan is probably the gibbon here, okay, because they have similar relative precipitation levels, again suggesting, and that gives us that indication of what their albumin is like relative to the human albumin. So in terms of exam phrasing, this would be the type of thing it would be looking for you to say, just that the chimp and the gorilla are more closely related because they show the same level of precipitation, and quote that actual answer at 95%. And then the nearest relative, the orangutan, is going to be the gibbon, as it only shows a 3% difference in the precipitation level, whereas all the other primates show a greater difference. So just pick out bits of that information from the table that you can use to make a comparison.